Alright, okay. So, hello, dears. Ayan, hello. Wait lang. Let me breathe. Okay, alright. Ayan. So, medyo sabaw ako ngayon, guys. No? Kulang sa tulog and medyo may asthma at asthma like, pero laban. Okay, alright. So, hopefully, dito ako kutasan ani. But anyway, hello, dears. Again, hello, good days, ninyo dira. No? Welcome to another pre-recorded lecture in our class in parasitology. And for this lecture, what we're going to discuss now is the main bulk, all right? And if not the most common, in a way, method no, of, or laboratory activity that we need to perform in the parasitology laboratory. Again, this is the bulk good of our work as medtechs in the parasitology lab. And that is the preparation of fecal smears, okay? Because again, as mentioned, diba, your stool is the most commonly submitted sample in the laboratory. Of course, so once it's already submitted to the laboratory, alangan, ato na siyang tutukan. Diba? So, di na natin siya tutukan. No? So, of course, once the stool samples have been submitted to the laboratory, diba, first, the first thing that we do is the macroscopic part. Of course, checking the label and all that. Yes, check your labels. And then, after checking is, diba, you examine the macroscopic part or the gross examination. That is what we have discussed in the previous lectures, right? So, after discussing or after examining the gross appearances of your stool for its color, consistency, and other pertinent constituents, no? Or materials there, like your proglottids, adult roundworms, if there are any. After noting those, then we proceed to making the fecal smears, or making smears, making slides out of your stool specimens to be examined under the microscope, of course. Because again, for us to really look, no, or for us to really identify what parasites are present in the stool sample, we need to make slides or smears out of it, all right? And that is what we're going to do, or what we're going to discuss in this pre-recorded lecture, okay? And again, the main bulk of our job as medtechs in the parasitology lab is to make, yes, fecal smears, okay? So, take note, this is just preparation pa lang ha. So how do we make the smears? Alright? Okay. Ayan. Now for a short introduction, the preparation of a direct wet mount, okay, that's another term, wet mount, is simple and efficient procedure for the examination of feces. So direct wet mount, later I'll discuss what it means, no? But again, when you say direct wet mount, is creating smears from your feces or from your stool samples. Alright? Okay. Because the mixing action of your intestinal tract, it concentrates in a way, or it makes the distribution of your parasites found in your system, in the intestinal system or your intestines, to be evenly distributed. All right, so you are to be expected, or you can expect rather, that in your stool specimens you can really see some parasites. All right, or you can see the parasites that cause the disease because of this mixing action. All right, okay. Next, wet mounts, by the name itself, it's wet, no? So you add a liquid, you add a reagent that is liquid. It should be examined immediately. Why? Because again, due to the reason that the liquid can evaporate. The reagent that you use can evaporate. So once the liquid has evaporated, then your examination will not be satisfactory anymore, or it would not be of diagnostic value, okay? It would not help you anymore because the stool itself is not already, cannot be visualized, okay? Because again, there's no liquid anymore that in a way suspends the stool, all right? Which to make the examination much easier, all right? Okay, so that's for the wet mounts. Again, immediately, all right? Okay, and several techniques if you want to preserve your smear, no? If you want to make sure, or like you want to look at your smears in the later period or in the later time or at a later time, you can use ringing the cover slip. So when you say ringing, ayan. So ringing, so this is your cover slip, no? So when you say ringing, you put sa sides, okay? You put sa sides either petroleum jelly or petroleum jelly and paraffin, or with nail polish, like atong colorless na nail polish, okay? So mga girls dyan, and even boys, na tig Q-Tix or tig nail polish, yes. The colorless na nail polish, yes, that can be used. So the main purpose of ringing the cover slip is so that the cover slip will stay put, okay? So that it will not move, okay? All right, so in a way, because the cover slip is already secured, all right, it's already been locked in a way by your nail polish or petroleum jelly, you can look, okay, you can look at your smear again at a later time. All right, because again, the cover slip has been already secured. Okay. All right. Now, before we go to the main discussion in the preparation of your smears, we'll look into some errors that you may commit after examination. After you examine a fecal smear. So first one is your epithelial cells and macrophages, which are again normal components of your stool samples. They can be confused with your amoebic trophozoites. So how do we differentiate? We look at the nuclei of your epithelial cells and macrophages because they can be seen in buffered methylene blue stain, and they appear larger and they may contain granules or particles. Example, this is an example of an epithelial cell stained, no? So as you can see, the border is not totally spherical, no? And it's quite irregular, and it has a large nuclei or nucleus, all right? Whereas for amoebic trophozoite, as you can see, the shape is quite spherical talaga, or like elongated. And as you can see, the nucleus is quite smaller, all right? Okay, ayan. And yeah, this is stained with iodine, that's why it's color brown. All right, and this time, this epithelial cell is stained, I think it's buffered methylene blue. All right, but again, look at the borders. It's irregular for epithelial cells, and the nuclei also are bigger, larger. All right, okay, so that's how you differentiate. The next error that you may commit are your pus cells or your white blood cells. All right, they can be confused with your amoebic cysts. So the nuclei of pus cells, they have 3 or 4 rings, and they stain heavily. The cytoplasm is ragged, meaning it's not smooth, okay, it's irregular, and the cell membrane is often not seen. For amoebic cysts, they have a distinct cell wall. So what am I talking about? This one. So this is an example of a cyst of Entamoeba coli. So as you can see, the cell wall of the Entamoeba coli cyst is definite. You can see the shape talaga na it's a circle or a sphere. Okay, and it's smooth, di ba? Ayan. Whereas for your polymorphonuclear, your PMNs, ayan, that's the meaning. Sorry. Your abbreviation, PMNs, polymorphonuclears, leukocytes, which are usually your neutrophils. Okay. As you can see, the cytoplasm is much dirtier in a way. And the borders are also not that clear, all right, or not that smooth. And usually they appear in clumps. Okay. And for Entamoeba coli, since it's a big amoeba, big ang kanyang cyst, all right, so it's much bigger than your white blood cells. All right? Okay. Bakit ako pinapawisan? Yes, medyo mainit pa dito sa room. But anyway, all right. So that's for pus cells and amoebic cysts. Okay, look at the nuclei. For nuclei of the pus cells, if you stain this, it's much heavy ang color. Whereas for the amoebic cyst, it's much lighter. All right? Okay, ayan. And again, look at the cell wall. Diba? Medyo definite, no? Circle talaga siya, spherical. Whereas for your PMNs or your pus cells, they are dirty. Or dili siya, it's not even. Okay? Ayan. Alright. Next, we go now to the hair and fibers. Ayan. We look at the internal structure because they may be confused with larva. So example, you have the plant hair in stool, and you have here a hookworm larva. So you can see, internal structure is much more complicated, alright, for the larva. Daghang sulod, okay, alright. Whereas for plant hair, as you can see, it's medyo clear ang inner, alright. And you don't see any parang mga internal structures, alright. So you look at that, that's how you differentiate, alright. So you may be asking, sir, why is there plant hair in stool or hair or fiber? Especially example for fibers, diaper fibers for infants, and diapers from your, fibers from your like clothing or something that could be introduced to your stool sample. For plant, siguro like during mga seasonal, maybe in the other and other countries that there are seasons, no? So there could be like mga spring, autumn, ganun, that could introduce you can introduce like pollens or like plant fibers that have been airborne because of the season, and then it lands on our stool sample, then that could be. All right? But yeah, or also from the food that you eat pala. Example, if plant-based siya, there are some plant fibers that cannot be digested by your intestines. All right? So they appear as fibers, or the fibers itself cannot be digested. So ayan. All right, look at the internal structure. For the larva, again, it's much more complicated. All right, and you can also see some larva, there's a mouth. Okay, all right, ayan. But again, for plant hair or fibers, simple lang ang inside. All right, there's not much more that's happening. Like parang walang masyadong laman. All right, okay, ayan. That's for hair and fibers. And next is, of course, plant cells, molds or yeast, all right, or your fungi, can be confused with eggs or cysts. Plant cells, thick ang cell wall, whereas ang cysts, thin walls. And yeast and molds, they are smaller than your amoebic cysts and do not have nuclei. Yes, they do not have nuclei. So you don't see any circle in the middle or something like that. Basta wala silang nuclei for your yeasts and for your molds, yes. Okay, so here's an example of a pollen grain. Diba? If you look at it, it looks like an egg of a helminth. Okay, all right. Pero this is a pollen grain. So how does a pollen grain arrive at your stool sample? Same again from other countries. There could be seasons, no? And pollens are really like example in seasons like spring yata, when there are a lot of pollens that could be airborne, yeah, because of pollination. So that could happen, that it could be introduced to our stool sample. So as you can see, diba, it looks like an egg, okay, of an ova. But in comparison with your amoebic cyst, ayan, so it's much more spherical, and there is always look at the nuclei, all right? So this is an amoebic cyst, and in comparison with an ascaris egg, because it looks like an ascaris egg, you're familiar with the ascaris egg naman, all right? So you have an embryo, and then you have the outer layer, all right? So just look at it. All right? Okay. And as you can see, diba, the cell wall of the pollen grain, if you look at your handouts, they're much thicker compared to the thin cell wall of your amoebic cyst. Okay? All right. Ayan, that's for pollen grain. And lastly, eggs of arthropods and plant nematodes may be mistaken as parasites. I have not experienced this, but if you encounter this, then you could always ask your pathologist for confirmation or from your other medtechs. All right? Basta just look at the parasites that we know, okay? Their appearances in their eggs and their larvae or adults and their appearances of their larvae or adults, all right? So they have distinct characteristics naman, all right? So if you can see other parang constituents that you think are eggs of arthropods or plant nematodes, since we do not discuss that, okay? Because again, we're focused on the nematodes or parasites that affect man, then you could always ask, for second opinion or for a second opinion from your colleague, like co-medtech, or from your pathologist if they are what you've seen is important, or maybe it's just an artifact. Okay, so usually these that we have mentioned, guys, these errors, these are known as your artifacts. Okay, all right, artifacts, or from Zybig also known as your confusers. Ayan, by the name itself, confusers or artifacts. They can be mistaken, no? They can look or they can appear like an egg, a cyst that could make your examination difficult. All right. So artifacts or confusers, they have no pathological significance, okay, in your stool sample. All right. Because again, they're just confuser artifacts. When you say artifacts, they are there, but they do not have any clinical significance. Okay. All right. Ayan. Now we go now to reporting of results. Generally, for reporting of results, when you examine under the microscope a fecal smear, it depends on one laboratory to another. They have their own standard operating procedures. But for our class, if under normal face-to-face classes, this is how we report. If protozoa, you see a protozoa in your stool sample, you report that as positive for the name of parasite and stage of parasite. So example, you see positive for E-hist, ayan, cyst, ganun. Okay, that's how you report. In our class, this is what we follow in our class, all right? Okay, if protozoa. And if it's absent, if you don't see any protozoa, none seen. Okay, all right. And lastly, for helminths, if you see a parasite, a helminth egg, then you count the number of the eggs per cover slip. So example, you see 1,048 eggs of ascaris per cover slip. So that's how you report. Okay? All right. Now you may be wondering, sir, how do we count eggs? All right. We use what we call this one. Unboxing, yes. A counter. All right. So you hold the counter like this. Okay. Actually, what makes you comfortable, no? like that, pwede. And then you click this for numbering. So as you can see. Ayan. So for every egg that you see in your smear, you then click this. Okay, so meaning if I click this, I see one egg of Ascaris, another egg, okay? Until you end, you finish the cover slip. Alright? So example 12. So it's seeing the number. And then if you want to make it back to 0, you just turn this. Okay? Ayan. Ayan. Until it reaches 0 again. So that you start from the beginning. Alright? So this is what we say, what we call a counter, tally counter. Okay, for counting the number of eggs. Alright, okay. Ayan. And if it's negative, you just say no ova seen. Alright. And this is an example of a sample form. This is found in your manual, supposed to be in our face-to-face classes, no? So you, again, describe the color consistency of your stool sample. Microscopic exam, are there protozoa that you see? Are there ova that you see? Okay, and then comments and examiner, means you. All right, okay, now, again, as mentioned, for reporting of results, when examining fecal smears, it depends on the laboratory. It depends on their protocols, okay? Example, in what I've experienced in Galeares and Soto, the hospitals that I have experienced my internship in, they do not count the number of eggs in ova, no? Maybe if it's too much, but usually what they report lang, or how they report it is, example, A. lubricoides, ova, seen, ganun lang, like that. That's how they report it. Or E. hist, E. dispar, positive for E. hist, E. dispar, cyst, or trophozoite. So that's how they report it. Okay, so they don't count the number of eggs per cover slip. All right, so again, that further supports my discussion or my notion that again, the reporting of results depends on each institution. All right, this is just what we follow in our class. Okay, all right, ayan. So that's for the intro lang, mga basics. Okay. Now, we go now to the different methods in preparation. Okay, so we'll start first with your direct fecal smear and your D'Antoni's method. These two usually are what we refer to as your wet mounts. Okay, so these are your wet mounts. Okay, and we have another last two. You have the Cato-Thick and the Cato-Cats technique. Okay, all right. Now, when you say wet mounts, okay, sige. All right. So, according to Bailey and Scott's, this is from Bailey and Scott's, guys, ha. Your ova and parasite, ONP, examination, it consists of three stages. Number one is your direct wet mount. Number two is your indirect wet mount. And the third is the permanent stain smears. Okay? All right. Now, your direct wet mount, it consists of the direct fecal smear, D'Antoni's. Some even perform Cato-Cats and Cato-Thick also. All right. Now, for direct wet mount, when you say wet mount, it's a preparation of your slide, of your specimen, where you use a liquid reagent. Okay? So, example, you have normal saline solution later, NSS, or iodine. Okay? So, what you're doing is you're suspending the stool sample in a liquid reagent. So, in a way, it serves as a wet mount. Okay? All right, that's for the first. Your indirect wet mount, before you make a slide, you first subject the specimen to concentration techniques, which is our next topic after this. Okay? All right. So when you say concentration techniques, by the name itself, you concentrate. Okay? So you subject your specimen to concentration. So what's the purpose? It's for accumulating, all right? Or like really gathering all the eggs, usually eggs, the ova that is found in the stool specimen. So, iyang i-gather, i-concentrate, all right? So that, again, you will see all of them in the stool sample, all right? So that you will know the intensity of your infection, especially if it's a light infection, meaning it's not too much, it's not that many of eggs yet, okay? So, you perform concentration techniques. So, that's an indirect wet mount. And by the name itself, permanent stain smears, which is again, a later activity in our class, we stain your fecal smears. Why? For visualization, especially for protozoa. All right, so we want to see, we stain. When you say stain, we put colors, all right, in our smears so that we can visualize our protozoa, especially the protozoa, much easier. Okay, so this is the 3 components now of your ova and parasite exam. That's according to Bailey and Scott. And most of your laboratories now, I think outside of the country, they don't perform the direct wet mouth na, like the direct fecal smear and all that, because it's not that accurate now, or it's not that sensitive, meaning you do not, you cannot examine everything, or you cannot usually get the parasite from your specimen, because again, it matters to one examiner, from one examiner to another. All right. So what they do is they perform concentration techniques. Okay. Because usually daw then the stool specimens there are preserved. Okay. They are preserved, so we cannot perform a direct wet mount that if it's already preserved. So they subject that to concentration techniques. Okay. All right. Ayan. So again, these are the different methods in preparing, okay, a fecal smear. All right, now for the next video, all right, so daming chika, no? We'll start first with the first type of preparation, which is your direct fecal smear. All right, okay, so I'll see you on the next video.